It's a great pleasure to be here and to share with you some of my passion for what this new era will mean for my patients. I treat a rare group of cancers called sarcomas, which are cancers of our connective tissue, like muscle and fat and bone. And they affect a young population in particular. And it's in the young that we think the things that we inherit are most likely when they get diseases young, that those factors are likely to be particularly important. But what I wanted to share with you today, I guess, is to put all of this, what you've just heard, into some kind of narrative, a narrative that we understand the history of mankind. So we're going to start off by disappearing back in time. And the reality is, something people don't think about much, is that cancer is a very ancient and fundamental disease. It kills about 1 in 3 in our community. Currently, about 40% of our community will be exposed to cancer, will develop a cancer of some kind. But it's been around for as long as humans have been around. This mummy, an ancient Egyptian mummy, was found to have a bone tumor, actually. The tumor that I treat, osteosarcoma, which affects young people, this skeleton had in the spine an evidence of a tumor thousands of years ago. And Hippocrates, the famous Greek physician, described cancers in the populations in ancient Greece. So cancer's been around and is as fundamental to us as humanity. In fact, it is an inborn defect in the way in which all of us are designed. Everything that is not a single cell has this fundamental ability to corrupt its code and be susceptible towards the growth of cells that parasitize us as hosts, which is what cancer is. Now, what I want to talk to you about is the way that we have thought about cancer over history. And I divide them into 3 ages. And in the first age, which really goes back before the Christian era, until about the middle 1800s, was dominated by an anatomic definition of cancer. And this is Andreas Vesalius, the great Italian surgeon, who in the 15th century used, for the first time, empiric studies of human anatomy to then form the fundamental principles behind surgery. If you have a tumor and you chop it out, you can cure cancer, and that remains true today, which is really important because there are ways in which the genomic research that we're doing will make surgery, the most fundamental form of treatment, more effective. This is John Hunter, a Scotsman and English surgeon, very famous in the 18th century, who for the first time started to develop the medical practice of surgery for the treatment of diseases like cancer. And this is an American, William Halsted, who did the first surgical procedure for a disease which we know affects many women in our community, breast cancer, and developed the first surgical procedure which we know today as mastectomy. And the reality is, although these approaches, the way in which we've thought about cancer as an anatomic entity, limited to an organ, a lump in an organ, as simple as that, is still relevant today. The second age of cancer was about the cell, and Marie has nicely described that we're all made up of cells, but it wasn't appreciated that that was the case until we had the equipment to look at it. And this is a really important, parenthetically, this is a really important principle about science. To understand new biology, you've got to have tools and technologies. And this microscope, the ability to look at a very small scale, the structure of things, was fundamental to the ability to see the cellular basis of not only us as humans, but of cancers in particular. And Rudolf Virchow here, these are some of the pictures. They didn't have cameras in those days, but they could look down the microscope, these brilliant people, and were able to describe the cellular structure that we recognize today. You can see the nucleus that Marie described in these cells. And in fact, when we look down the microscope today to look at cancers, and this is a sarcoma from a patient that I might have treated, you can see that the cellular structure here looks quite chaotic. Now, normal tissues don't look chaotic, and it was recognized that cancers were formed of these chaotic aggregations of cells, which that was how we defined cancer in the second era. And we still use that today. So right today, if I saw this appearance under a light microscope, it would change the way I treat a patient based around what is effectively almost art. It's a subjective thing, but we're beginning to get closer to what the origins of cancer are. And this is effectively now the third great era of cancer. And Marie mentioned these two individuals, but there are two others, Maurice Wilkins and Rosalind Franklin, who contributed to this fundamental paper, groundbreaking, one of the great papers in modern biological science, published on April 25 in 1953, in which DNA was described in its structural form. And here's, this is Jim Watson, and that's Francis Crick, in front of a model that they designed that illustrated for the first time this microscopic structure of the chemicals of DNA. And that's been fundamental, because at that point in time, we began to have the tools to be able to now look at the molecular nature of cancer, the third age of genes and molecules. And that gave rise to some fundamental concepts. And so the way that we think about cancer today has been nicely elucidated in this paper in the middle 1970s by a man called Peter Noel. Peter Noel described the first mutation in cancer, along with David Hungerford, the Philadelphia chromosome, which is seen in chronic myeloid leukemia. And that's a recognizable thing that you can see amongst the chromosomes of chronic myeloid leukemia cells, these rearrangements. The first thing we could actually see, we had to use our eyes and a microscope to see it, but we could see it because it was as big as a chromosome. Whereas the sort of changes that Marie's been talking about in single nucleotides were invisible at that time. And this gave rise to a concept which I think applies today, that you take a normal cell and then some change occurs. This is chronic myeloid or chronic granulocytic leukemia, and a change occurs here to introduce that mutation. And then the mutation causes the cell to have a growth advantage, and it starts to divide, and it acquires subsequent mutations in what is called a clonal fashion. And what that means is that a single cell gives rise to all these changes, which eventually reaches the point where it starts to grow in a completely uncontrolled fashion, and will ultimately cause the death of the person in whom this develops. And this concept of the cellular basis of cancer linked to a mutational concept of cancer is still present today. But while we could describe this at a certain level, the ability to apply this to all cancers, all of the 120,000 Australians who every year will develop cancer, has not been possible until recently. The technology to do that has had important implications. This is what we call, this is really state-of-the-art science, so I'll just take you through this. This is now a representation of chromosomes. You can see here the X chromosome up here, the Y chromosome, 1, 2, arranged in a circle. And you can see, remember those bands on the chromosomes that you saw before? These are the bands in the ideogram of those chromosomes. And these changes in here represent the rearrangements of the normal DNA in the cancer cells of a patient with melanoma. You can see this is a PET scan, which is a way of imaging cancer in a person. You can see there's lots of cancer. This is normal brain, that's nothing to be concerned about, but this stuff is all bad. Now what we've been able to do is take this information and then work out what's broken to cause the cancer to grow, one of those key changes that you saw earlier that allows the cancer cell to grow and form a mass, and then treat it with a drug. And this is an example of the drug vemurafenib, and a clinical trial of 275 people who received vemurafenib. And this is a melanoma patients, 274 of them who received what was then only a matter of a decade ago, the best treatment that we had, only 10 years ago. Remember, just pan back for a second, thousands of years, in the last 10 years, a blink of an eye. This treatment, based around a molecular map of that particular person's cancer, has enabled people to go from a median survival of 6 weeks out to almost 6 months. That's radically changed the possibilities for those people. And this is, it doesn't matter what you show in amongst 500 people who are treated this way, but this is what it meant for that one person. And what that person is now able to do is get out of a wheelchair, stop taking opiates, get value out of life. So this is a miracle that we see every day in the clinic. We see the reality of this technology being used as proof of principle that we can change what happens to people. I'm going to talk about another aspect of this. So when the cancer gets to look like this, unfortunately, it's too late. And I've mentioned surgery being critical. What we want to do is pick up a cancer when it's like this, before it grows to that. Remember that growth curve, the way in which the cells expand? They start off as something that's small like this. We know if we get it at that stage, the surgeon can take it out and cure people. And still, the majority of cures that occur in cancer today are because of the surgeon being able to get the tumour early at that stage. But how do we get there early? Because this tumour, this is a lung cancer in this particular case, is completely asymptomatic. The person with that cancer will not know that that's there. How do we pick it up? Well, the reality is that cancers don't evolve out of the blue. Those cancers that we've been describing, those mutations, occur in the context of a person, and that person has a different genetic code, and that genetic code is embedded in families who may get cancer. And of course, all of our society, all those faces that Marie described before, are actually woven into a web. We're all connected. We're all related to each other in some complex way, and some of us are related more closely through our family structure. The question is, what has genomics got to tell us about the population at large who has cancer, and can we use that information to work out who has a cancer before it spreads? And that, I think, is what the future will look like. Now, we've known for a long time that cancer is an inherited disease. This paper came out in 1932, but it was a description of a family, and the black dots are people who've got cancer within this extended pedigree in this family. But the paper itself was described originally in 1911. It's ancient. This is fundamental as cancer itself. And we know about cancer affecting women who carry the BRCA1 and BRCA2 gene mutations that you might have heard about, where the mother passes on to the daughter that gene, and that gene gives the daughter up to a 90% lifetime risk of developing breast cancer. And we know we can do something about that. The question is, how do we go from these rare, exceptional families to understanding the patterns of cancer in the faces in that crowd? Or in this room, all of us have a completely different risk of developing these diseases. And I believe that we'll be able to use genetic information to work out who's at risk and who's not. And just to consider what that might mean. Currently, in this country, we spend $120 million each year doing mammographic screening on every woman over the age of 50 for breast cancer because it's such a common disease. Now, for some of those women, there is absolutely no risk of getting cancer. We know that, or very low risk at any rate. And so those people probably don't need to undergo screening to that sort of intensity every 2 years from the age of 50. And there are another group of women who are at very high risk of getting cancer well before the age of 50 years. We can use this information to start to personalize our screening programs and thereby potentially invest the same amounts of money more effectively to help more people and avoid the burden of screening upon those who will not benefit from it. And just to show you what the benefit is, I love this slide. I'll just take you through it slowly. This is the percentage of people who are alive following a diagnosis of breast cancer. One is everybody's alive, and this is time, so of course over time people go on to die. And this bottom line here is the group of individuals in the UK, thousands of them, with women with breast cancer between 1958 and 1967, who were not subject to mammographic screening. This is the next cohort. You can see a slight improvement in outcomes because of improvements in medical care, who are also not screened. And then this is a cohort between 1978 and 1997 who were also not screened, and you can see still further improvements as our treatments have got better. And this is the group of women who got breast cancer who were subject to screening. And what that shows you is that early detection using technologies of this kind has had a more massive effect on survival from breast cancer than anything we've done in medicine over that period of time. It goes to the point that if we screen effectively and detect cancers early, we can save lives. Of the families that we understand about risk, currently we think it applies to around between 5 and maybe 10% of all cancers, but we've focused only upon 23% of cancers. So 75% of cancers, roughly speaking, have never been studied in this way. This is the bottom of the iceberg. As we apply these technologies, because their prices drop across the entire population, we'll discover that probably between 1 in 10 and 1 in 4 people in this country, that is 10 to 25,000 people a year, will have some marker that we can identify that points them, and of course their first-degree relatives who don't yet have cancer, as at risk. And that's a really important public health opportunity that we in the Cancer Division are focusing on going forward. And of course, that's all dependent, again, just like the light microscope, upon technology. And we're right at the cutting edge of this space. We have this incredible technology that can look at whole genomes at the population level. We can ask questions about, now for the first time, about public health issues using these technologies. And it will give us not only insights into what we can study to date, which are the coding sequences, which amount to some 2% of the genome, but the remaining 98%, which has been dark matter, and where I think a lot of variation is going to be revealed that will account for many diseases. And this just puts another element to this. We've gone to that, from that mummy, in this short talk, to where we are today. We need to do this, a petascale computer. We need to have something that is capable of doing 1.2 petaflops of processing to handle this. That is a million, a billion, 1,200 billion processes per second to analyze this data. We need 40 petabytes, not terabytes, not gigabytes, but petabytes of data space to do this. And fortunately, in Australia, we have a supercomputing facility which has been developed not for biology, but in fact for the Bureau of Meteorology and CSIRO to handle astrophysical quantities of data, but turning for the first time, thanks to a partnership with the Garvan, into something that will be focused upon deconvoluting this complexity for the purpose of treating patients. And a piece of data I love about this is the power consumption of a computer is 2 megawatts. You need to have an entire cooling system to handle the heat generated from the processing of that data. In the UK, the 100,000 Genomes Project is building its computer over a mine shaft in Wales so that the natural cool air from the mine shafts will cool down the computational stuff. Isn't that freaky? Now, I want to finish off my short story with telling you about where science is taking us and evidence that it's of the improvement. This is UK data about changes in survival from cancer between 1971-72, when they first started collecting good records, to about 2010-11. And this is for all cancers. We've gone from a 25% incidence, we're actually picking up more cancers. Sorry, 25% survival overall to a 50%. We've doubled the survival of cancer simply with the treatments that we've had over the past 40 years. And you can see for some cancers, thankfully, we're getting close to 95, almost 100% cure rates. Even for melanoma, we're picking these things up earlier and treating them more effectively. For some, very sadly, we've got an enormous amount of work to do. And I hope pancreas is down there because that's one of the signature projects of the Garvan, because it remains a dismal disease with a 5% 5-year survival, and that hasn't changed over the decades. But what I hope you can see is that intelligence and commitment and investment in technology and research makes a big difference to what happens to all of us. And I think the next era is going to be even more exciting than this. Thank you.